I'm going to be talking about the neural changes that occurs in the brains of people with Alzheimer's disease. I recently wrote a research paper in this field, and I'm glad to be here today to share it with you guys. So if you have any questions for me, you can just pop them in the chat. I don't think I can see the chat right now, but I'll answer them at the end. And if you have any additional questions, you can just email me. Okay, so as I said today we're going to be talking about the structural and functional changes of Alzheimer's disease. And these are like the main topics that we'll be covering. First, we'll be talking about what Alzheimer's disease is. Second, we'll be introducing MRI and PET scans. And then third, we'll be talking about the specific structural changes and fourth, the functional changes. So before we get started, if you guys are interested in reading my paper, you guys can scan this QR code right here. And I think Eliza will also post the link to the paper in the chat. Okay, so let's do like a basic introduction into Alzheimer's. Alzheimer's is a progressive neurodegenerative illness common in elderly people. That means that the disease has many different stages and it is a condition that worsens over time. And as you can see on the slide here, it's a senile dementia. It's a type of senile dementia. So it destroys memory and other important mental functions over time, and it gets worse over time. So eventually, at the very last stage, the patient has difficulty completing even just daily tasks like brushing your teeth or like washing your hands. So what actually causes Alzheimer's disease? These are some pathological causes that scientists have known to cause Alzheimer's disease. And we know that it's a buildup of two proteins called amyloid beta and tau protein. And the accumulation of these proteins can cause plaques and like tangles to form in the brain. And then these tangles can cause neurons to block, and they impede the communication from one neuron and another neuron. And eventually these neurons will die. And that's how Alzheimer's kind of come about. So how do we diagnose Alzheimer's disease? So now we're going to get into more of the technology aspect of how to diagnose Alzheimer's disease. So the first method that people use is MRI. And MRI just takes pictures of organs in the human body, and they uses radio waves and strong magnetic fields to produce these images. So they'll roll the patient in, and then they'll take the image. And there are two different kinds of MRIs. There's structural MRI and functional MRI. So structural MRI lets us see the anatomical structure of the brain, and in Alzheimer's, they let us see the structural changes that occurs in this disease. And functional MRI lets us see brain activity by detecting changes associated with brain flow. And that's going to detect some of the functional changes in Alzheimer's. And these three scans are what MRI scans look like. If you can see, the first scan here is an exile scan. It's like a scan of like the top of the brain, and there's one middle here is a sagittal scan, and it shows the side of the brain and kind of the spine. And the one on the right is a coronal scan, and it shows the back of someone's brain and also some parts of the spine. So another way we detect changes in Alzheimer's disease is using a PET scan. So PET scan, how people use PET scan is that they inject a small amount of sugar into a vein, and this sugar, they will review the metabolic function of tissues and organs, because where your brain uses is used the most, where there's more brain activity, the sugar will also be consumed the most. So using PET scan, they'll see which part of the brain is most active and which part of the brain is not active. And this can be used to trace diseases and the parts of the brains that are damaged in diseases like Alzheimer's. So these are some examples of PET scans. As you can see here, the red part is very high activity, and the blue part is very low activity. So that's how scientists kind of distinguish where there's high activity and low activity in different parts of the brain. So this picture right here is a comparison of MRI and PET scan. So MRI looks like this. Structural MRI looks like this, and functional MRI looks like this, and then PET scan over here looks like this. So PET scan tends to have color, and that's how, that, as I said, people distinguish brain activity in PET scans. And structural MRI and functional MRI is right over here. So what has structural MRI detected in Alzheimer's brain? So now we're going to get into the structural changes, like we talked about in Alzheimer's disease. So people have detected brain atrophy in Alzheimer's patients, and brain atrophy is just basically a shrinking in the tissues of a part of the brain, such as like the hippocampus or entorhinal cortex in the brain. And this atrophy can be generalized or focal, which means it can be the whole brain or it can be just a specific part. And it can lead to a loss of neurons and a loss of connections between these neurons, which means this will cause a certain part of the brain to die out, and a function of the brain can no longer be used. So how is brain atrophy displayed in Alzheimer's? So brain atrophy is, like I said, a shrinking of a part of a brain, or a whole brain. But in Alzheimer's, it's more a part of a brain, and it's in the medial temporal lobes and in regions such as hippocampus and entorhinal cortex. And how do people know that brain atrophy is severe in Alzheimer's? Is that the rate of atrophy in Alzheimer's is about 1.4 to 2.2 in Alzheimer's annually. And in just normal aging, it's only about 0.7. So as you can see, the rate of atrophy in Alzheimer's is significantly larger and more severe. So now we're going to get into specifically hippocampal atrophy. So if you look at the picture at the top, you get an idea of where the hippocampus is. And just a little fun fact about the hippocampus, it actually got its name from seahorses because its shape and form is almost like a seahorse, as you can see from the picture below here. And according to many studies, scientists have been able to deduce that atrophy in hippocampus is present a decade before appearance of Alzheimer's symptoms, such as memory loss and other symptoms such as difficulty comparing tasks and just like that. And as the diagnosis of Alzheimer's nears, scientists also noticed that the shrinking of the hippocampus progresses, which means it gets more severe and severe as the patient's cognitive functions decline. So for example, as a patient gets closer to being diagnosed with Alzheimer's, the hippocampus shrinks severely, and the memory functions of the patient can also get worse and worse as this atrophy gets more severe. And if you guys know already, the hippocampus is mainly responsible for memory loss, and the hippocampus is mainly responsible for memory storage and retrieving memories. And since memory loss is one of the first symptoms of Alzheimer's disease, then scientists know that Alzheimer's patients, they can correlate this atrophy with Alzheimer memory loss in Alzheimer's disease. And scientists have also noticed that there's greater right hippocampal atrophy than left hippocampal atrophy, which means the right side of the hippocampus is more severely damaged than the left in most Alzheimer's patients. So these are hippocampal atrophy scans in Alzheimer's patients. As you can see in this A scan right here, the circle part is the hippocampus. And you can see that it's very tiny and it's very weird out in the A scan compared to the B scan. In the B scan, it's fuller, and in the A scans, it looks like it has shrinked a lot. And that's a clear example of how scientists can distinguish hippocampus and atrophy in Alzheimer's patients. So now moving on to entorhinal cortex atrophy. Entorhinal cortex is the most heavily damaged cortex in Alzheimer's disease. So people with the APOE4 gene has also been deduced by scientists that they have great entorhinal cortex volume loss before the diagnosis of Alzheimer's. And people with the APOE4 gene, just to clarify, they're at great risk for Alzheimer's. So scientists are able to deduce that entorhinal cortex volume loss is correlated with this gene. And how is the entorhinal cortex related to the hippocampus? The entorhinal cortex is connected to the hippocampus because it gives rise to axons that interconnect with the formation of hippocampus and the rest of the cortex. So this is a model of Alzheimer's progression. So if you look at the years from Alzheimer's diagnosis, as the years go past and go near Alzheimer's diagnosis, the atrophy in the brain also increases with the impairment in the brain. So impairment means there's a decrease in cognitive function. So as atrophy increases, the cognitive functions decreases, which means as a patient, for example, a patient has greater hippocampus volume loss over time, the patient also has severe memory decline over time. And this is just another picture of what brain atrophy in advanced Alzheimer's disease looks like. As you can see from the normal scan right here, it looks full, like the brain doesn't look so weared out and shrinked. And in the Alzheimer's scan right here, the brain obviously looks very abnormal. And these two bottom scans right here are also very good examples of atrophy in Alzheimer's versus a normal brain. And now we're going to talk about ventricular enlargement, which is also a structural change in Alzheimer's disease. So what are ventricles? So ventricles are these four hollow cavities in the middle of your brain, and they're usually filled with CSF fluid. And CSF fluid protects your brain and your spinal cord from trauma, and it also transports nutrients to the cavities. And this is what the ventricles look like on an MRI scan. They look like an X, as you can see right here. So Alzheimer's ventricular enlargement reflects the expansion of cerebrospinal fluid into regions occupied by living neural tissue. And this just means that as the ventricles get larger and larger, the fluid in these regions also expand and into these living tissues where they usually are, and this can cause damage to the brain, and we're just going to talk about that later. And these ventricles expand by about 16% in Alzheimer's patients annually, and only about 1.5 to 3% normal elderly patients. And this means that in Alzheimer's patients, the ventricles expand a lot in comparison to just normal people, and we can see that through like MRI scans, and that's how scientists know that there are enlarged ventricles in Alzheimer's patients. and compared to subjects with mild cognitive impairment, which is a disorder that can lead up to Alzheimer's. Um, People with Alzheimer's has a 60 greater ventricular enlargement over a 6-month period, which means that they have significantly greater ventricular enlargement over the certain time period compared to people with another disorder, which means Alzheimer's is ultimately more severe and is a resulting cause of mild cognitive impairment. And right here is a scan of a patient with Alzheimer's disease. As you can see, the patient has very enlarged ventricles in comparison to the normal patient, which has very tiny ventricles. So that's just a real life example of how scientists can see the enlarged ventricles. Right here on this slide is the systematic scans of all of the ventricles. So ABC scans show the ventricles of normal elderly. And as you can see, they're very tiny, but over here in the D scan, the ventricles are very large. And that's just another example, like the slide before. So this is a complete model of the structural hallmarks of Alzheimer's. So as Alzheimer's disease progresses, the tissue also shrinks. And as you can see from the images right over here, the ventricles also enlarge. And this can cause just cognitive functions changes like memory loss and difficulty speaking and all that. And so when the disease spreads through the cerebral cortex, language, judgment, behavior, and bodily functions decline with memory. And this can just lead up until death, which is the most severe stage of Alzheimer's. As you can see on the right over here, the ventricles are very enlarged and the hippocampus is very shrinked. And this is the last stage of Alzheimer's. So now we're going to talk about the functional changes in Alzheimer's disease. So the first thing we're going to talk about is the default mode network. And that's a network of regions that lights up when someone is not focused on the outside world and instead is focused on the inner world. That means that, for example, someone is sitting at a doctor's office and they're thinking about what they're going to eat for dinner. But for example, if someone is doing a math problem or something, the default mode network would not be responsible for that because they're doing a specific task. A default mode network is more when someone's just thinking about their life and just their life purpose in general. And this network is especially active when someone's not involved in any specific mental activity, like I said. So some general changes in the default mode network. So the posterior cingulate cortex and the inferior parietal lobes are most commonly affected because they're a part of the default mode network. So scientists can see that when people have Alzheimer's disease, these parts of the brain do not light up and they're consistently show across many Alzheimer's patients. So they also see that disruption in the default mode network is responsible for disruptions in episodic memory processing, which is a long-term memory. So episodic memory processing is the ability to kind of recall past memories. And scientists see that disruption in the default mode network is correlated. As I said, Alzheimer's participants also revealed a lower hippocampal coactivity within the default mode network. And this actually correlates with the episodic memory processing because, as I mentioned before, hippocampus is responsible for memory and retrieving of memory. This means that within the default mode network, the hippocampus does not light up as much in Alzheimer's patients. And lastly, the presence of the gene also the APOE4 gene also correlates with default mode network changes. And as I mentioned before, the APOE4 gene is the gene that's responsible for familial Alzheimer's disease. And these changes were present in frontal and posterior areas and the right hippocampus. So some things about the pathology and the default mode network changes and how these two correlate with each other. So tau protein has actually been found to accumulate within the default mode network, which means that as the tau protein increases and increases in an Alzheimer's patient, the default mode network is more severely impaired. And scientists don't know why this is. Like I said, scientists also still have a lot of like unanswered questions about Alzheimer's disease, and one of them is about default mode network. They don't know why these changes occur. They just know that they're present in the brains of these Alzheimer's patients. They also see that as amyloid beta protein increases, regions outside of the default mode network also become severely impaired with regions within the default mode network. This means that amyloid beta protein is correlated with this network activity and is impaired in Alzheimer's patients. So now we're going to talk about hyperperfusion in Alzheimer's disease and just what hyperperfusion is in general, because I know that's like a super technical term. So what is brain perfusion? So brain perfusion is the blood flow to areas of the brain, and the areas of the brain that are very active often show greater brain blood supply. So as I mentioned before, PET scans can actually detect these brain activity, but we're going to use perfusion MRI to use these to detect this hyperperfusion in Alzheimer's disease. So hypoperfusion, hypo means below normal. So we can kind of see that hypoperfusion means inadequate brain blood flow. So this is a comparison of the blood supplies in areas of the brain in Alzheimer's patients versus in normal control patients. As you can see right here, in normal patients, there's a lot of brain activity in all regions of the brain, but in Alzheimer's patients right here, there's only a tiny little parts like scattered all over the brain. So hyperperfusion is present in almost more than 60% of Alzheimer's patients. So it's something that's a majority of Alzheimer's patients, and it's consistently shown. So there's a dramatic global decrease, like more than 40% in blood flow compared to healthy controls, which means that in Alzheimer's patient, there's a significant decrease in blood flow than in just normal control patients. And there's also a reduced blood flow in regions such as the parietal lobe and posterior cingulate cortex. And that's just where the scientists have detected that this hypoperfusion is most prevalent in regions with gray matter loss or atrophy has persistently shown diminished blood flow, which means that atrophy is actually correlated with hypoperfusion. And that's something that scientists still have to study and gather more evidence on because, as I said before, scientists do not have all the answers to Alzheimer's disease just yet. So this is a perfusion MRI scan right over. All these are perfusion MRI scans actually, and they're just different views of these scans, and they just let you this is just an example of the scans of Alzheimer's disease.